Hey, what's up, guys? Welcome to the last minute CRISPR review video. I'm Chris. Today we'll go over the history of CRISPR, how transcription and translation differs in humans and bacteria, and how CRISPR is and can be used to apply genetic information in future applications. Oh, also we're also going to be going over transcription and translation real quick, so you understand what CRISPR is actually doing. So, transcription and translation. Transcription, it is the process in which DNA gets transcribed into RNA to be sent to the ribosomes, and then translation is when RNA gets turned into proteins that can make changes either in or out of the cell, depending if it's multicellular or not, and the function of the protein itself. So, transcription. So essentially, transcription. First, you have the RNA polymerase. This unwinds and then rewinds the DNA for you and allows the RNA to actually be translated. So it actually binds to the template strand. There's two strands, template and coding. The coding strand is essentially the exact same as the RNA strand, except that the T's are replaced with U's. So this is like essentially the same sequence. But since you need to bind, you use the template strand instead. So the RNA like binds and then creates essentially the opposite, which is why you use the template strand and not the coding strand. And that creates your RNA. It's pretty cool. But before it can like actually get there and everything, it needs to go to a promoter. Promoters differ between uh, eukaryotes and prokaryotes, but most eukaryotes use a TATA box, which is just a sequence of thymine and adenines before to know where to go, and then it also has like a ending as well. Now, there's a lot more stuff going on here as well. Essentially, this is also okay. So your RNA is being formed here, right? But it's not mature yet. It's not an mRNA. That's not that stands for, but um, so when you make this RNA here, right, you're essentially trying to send it outside of the cell, of the nucleus. It needs a little bit of help to do that. So first, you want your important stuff actually going out of the cell because we already know what a codon is, right? Codon are three segments of DNA here. Like AUG would be one, AUC would be one, UCG, and UAA, right? Until we get to a stop codon of some sorts, right? So we need to take out the introns and keep in the extrons. A good way of thinking about it is like since it's in a nucleus, right? The extrons go out of the nucleus and the introns stay in the nucleus. So that's why we have so much genetic diversity too, because bananas have more DNA than us, but they're obviously a banana and we're humans. One of the biggest factors that like leads to our genetic diversity is how many proteins we can make from just like a single gene line. Because we can take out multiple intron and extron combinations, a lot more than other organisms. That's what makes us unique and cool. Anyway, that's called RNA splicing, where those are taken out. And then before he can graduate, right, become a mature RNA, um, he essentially gets like a little cap and a poly A tail, which is like chain of four or more uh, tail adenines added on, so it can survive the journey from the nucleus to the ribosome. And that is essentially what transcription is. Now, translation, on the other hand, is the process of turning mRNA, the RNA strand we just made and moved to the ribosome, into protein. So the mRNA goes to the small subunit of the ribosome and gets read there, and the large subunit, which actually makes the protein with the help of the tRNA or transfer RNA, this guy right here, where was I? And the anticodons on it. So the anticodons are essentially just like what we did before in transcription, where there was opposite base pairing here with the RNA and the template strand, but instead it's anticodons with the codons. And those typically have an amino acid attached. And the sites that you see here, E, P, and A, are just different holding sites for the tRNA. First, the A site is where tRNA enters the ribosome and binds their anticodons to codons to allow for actual protein synthesis to occur. The P site kind of just holds the tRNA that is currently carrying the polypeptide chain while it's still growing, and it adds an amino acid whenever the tRNA that was in the A site decides to move over to the B site. The E site, or like exit site, as some people call it, is where the tRNA exits the ribosome after they do their job in protein synthesis, and a bit of forming and a whole lot of steps later, that makes a protein. It has to stop eventually at like a stop codon like UAG, UAA, or UGA, but that whole process is what makes a protein. When you have a mutation that like changes that UAA or UGA or UAG, it can lead to like a longer misformed protein that doesn't like do anything or does something even worse. Kind of like sickle cell anemia, where it turns one of the, I believe, guanine. No, it changes like a G to like an A or something, and that changes up one of the proteins in the chain. So it makes one of the molecules that wasn't polar, and that like crunches the shell of the red blood cells, which creates that sickle shape. So now that we know about these, let's talk about the differences between human and bacterial cells real quick. So essentially, origin point on this guy, because their DNA doesn't have a nucleus, they do it a lot easier. They do transcription and translation at the same time, and we don't. That's basically the entire gist of it. And they also have an origin point, and the DNA is circular and easier to penetrate. So Cas9 and the history of CRISPR real quick. So Cas9 is the bacterial protein that can defend the bacteria from bacteriophages, and was first discovered by these two people in Spain. They also developed the CRISPR that we use currently for genetic engineering. Pretty cool. So Cas9, main topic of this video here, very cool dude. But he's also kind of dumb. You have like that smart friend that kind of knows what they're doing, but then at the same time, like they're really smart, but then they don't have street smarts. That's basically Cas9 right here. So he's essentially a surgeon, but he doesn't know where he's cutting and how to get there. That's okay. We have buddies to help him get there. The guide RNA and the PAM sequence. So this gRNA or guide RNA helps Cas9 actually get to the site on the DNA that we want him to cut at, right? And Pam also helps him. He's like, "Hey, buddy, CRISPR, cut over here, guy. I'll be like your helper promoter guy over here." And he's like, "Oh, okay, I'll just cut here. Just Roger, Roger." And he just makes a nice clean cut down the middle, right? Oh yeah, it's right here it shows it shows everything, right? Makes a nice clean cut, and then the actual like genetic engineering part of it essentially is the repair template. So your body is already naturally good at repairing everything, but this repair template is just another thing that we put in with Cas9 that can hopefully initiate a genetic change. It's kind of like gambling, really, because like evolution, it's kind of everything's kind of lazy. It'll do the bare minimum, like finding free floating nucleotides. We're hoping it finds this specific nucleotide that we put in to match up and then create an actual genetic change. But it's basically just gambling because it could use free floating nucleotides too, which elicits no change, or it could like change it, cut it in a different sequence. So a lot of the CRISPR technology right now is trying to focus on making better guide RNAs and PAM sequences, so Cas9 can actually get to its right place. But yeah, the DNA essentially repairs itself. Pretty cool. Now, a little bit of history of genetic engineering: flavor saver tomato, glow in the dark zebrafish. Two really cool stuff that happened, but before 2013, we essentially couldn't genetically engineer stuff unless it was in an embryo or we just threw radiation at it. So clearly not the best ideas here, but we did get these two cool products from it. The coolest thing about CRISPR is its future applications, like getting rid of any drought. Like we can make plants more drought resistant, and we can also make people not have genetic disorders like diabetes. It's pretty cool stuff. I have a lot more written in my script, but I don't have the time for that. Have a good rest of your day, guys. Hope this helped.